Introduction. The word insect derived from the Greek word insecare, means cut in pieces or engraved, segmented. Insects belong to the phylum Arthropoda, artho equals jointed, poda equals legs, which is the biggest phylum of kingdom Animalia. More than 3/4 of the animals on Earth are arthropods, and most of these are insects. Kingdom Animalia is classified into 12 phyla. Insect. The insects are the trachy arthropods in which the body is divided into head, thorax and abdomen possessing 2 pairs of wings and 3 pairs of legs and single pair of antennae. Entomology. Greek word entomon equals insect. Logos equals study. It is the branch of zoology or biological science that deals with the study of insects. Branches of entomology. 1. Study and use of insects in crime investigations is known as forensic entomology. 2. Study of insects related to livestock and veterinary animals is known as veterinary entomology. 3. Study of insects in relation to human beings is known as medical entomology. History of entomology. In ancient scripts like Ramayana and Mahabharata, some of the terms used were related to insects. They are Pipilika, ant. Pathanga, grasshoppers. Madhumakshika, honeybees. Ambakapalika, termite queen. References of insects in ancient Indian literature. Mahabharata, 1424-1366 BC, mentions about silk, honey and lac. The famous story of Lashgrua, i.e. House of Lac, built by Kauravas to burn their cousins live, Pandavas. Amarkosha Sanskrit dictionary provide references like patanga and brahmar of flies, moths, beetles and glowworms. Sushruta, Surgeon 100-200 add classified ants, Pipilika, mosquitoes and flies. Umaswati, Physician 100 add classified the bees. Pests of national importance, e.g. locust, termite and white grub. 3. Contribution of scientist. Aristotle 384-322 BC. He was the father of biological classification. First person grouped insects in mandibulate and haustellate types and winged and wingless groups. He gave the terms like Coleoptera and Diptera. Carolus Linnaeus, 1758. Father of taxonomy. In his publication, Systema Naturae, 1758 includes 28 Indian insect species. J.C. Fabricius, 17,451,808. Danish entomologist described over 10,000 insect species. Published, Philosophia Entomologia, in 1778. World's first textbook in entomology. The first entomologist who made any extensive study of Indian insects. Classified the insects into 13 orders based on type of mouth parts. J. G. Koenig, 1767-1779. J. G. Koenig was the first to collect number of insects from Coromandel in southern India. For his memory, red cotton bug is named as Dicercus koenigi snodgrass R.E. 1875. Great insect morphologist, referred as a father of insect morphology. He wrote book Principles of Insect Morphology. Lionel de Nicevel, 1901. Was appointed as the first entomologist to the government of India. Maxwell Lefroy, 1903. Was appointed as the second entomologist to the government of India. First imperial entomologist. 1906 publication of Indian Insect Pests Up. 1909 publication of Indian Insect Life, Mithun Lal Roomwal, 1908. Outstanding work on termites, white ants. Contributions to ecology, embryology and locust study are noteworthy. Sir Vincent B. Wigglesworth. Insect physiologist, work on blood-sucking bug, bed bug. Father of insect physiology. T. B. Fletcher, 1914. The first government entomologist of Madras State. Wrote a book, Some South Indian Insects. Second imperial entomologist. 4. T. V. Ramakrishna Iyer, 1940. Wrote a book, Handbook of Economic Entomology for South India, H. S. Pruthi, 1963. 1934 First Indian, Imperial Entomologist of Independent India. 1963 he become first Plant Protection Advisor to Government of India. Wrote a textbook of Agricultural Entomology, Dr. M. S. Mannies, 1968. Wrote a General Entomology. Dr. S. Pradhan, 1969. Wrote a Insect Pests of Crops, and father of modern applied entomology in India. B. Vasantharaj David and T. Kumaraswamy, 1975, wrote Elements of Economic Entomology. M. R. G. K. Nair, 1975. Wrote Insects and Mites of Crops in India. K. K. Nair, N. Ananthakrishnan and B. Vasantharaj David, 1976, wrote General and Applied Entomology. 1912 Plant Quarantine Act was enforced. 1914 Destructive Insects and Pests Act was enforced. DIPA. 1916 Imperial Forest Research Institute established at Dehradun, Uttarakhand. 1925 Indian Lac Research Institute started at Ranchi, Bihar. 1937 A laboratory for storage pests was started at Hapur, UP. 1937 Entomology Division was started in IARI, New Delhi. 1939 Locust Warning Organization established. 1946 Feet Directorate of Plant Protection, Quarantine and Storage, of GOI started. 1968 The Government of India enacted Central Insecticide Act, which came into force from the 1st of January 1971. Establishment of entomological institutes. IOBC, West Indies, Trinidad, International Organization for Biological and Integrated Control of Noxious Animals and Plants. ISAPE, Kenya, International Center of Insect Physiology and Ecology. NCIPM, New Delhi, National Center for Integrated Pest Management 1988. PDBC, Bangalore Project Directorate of Biological Control -1993, and then PDBC or NBAII, National Bureau of Agriculturally Important Insects 2009, and Nowhere National Bureau of Agricultural Insect Resources. CIB, Faridabad, Central Insecticide Board. NPPTI, Hyderabad, National Plant Protection Training Institute 1966. IGSI, Hapur UP, Indian Grain Storage Institute. 1946 Directorate of Plant Protection Quarantine and Storage DPPQS Faridabad. 1937 Establishment of Entomology Division at IARI New Delhi. CABI, UK, Commonwealth Agricultural Bureau International 5. Economic importance of insects. The field of entomology may be divided into two major aspects. 1. Fundamental entomology or general entomology. 2. Applied entomology or economic entomology. Fundamental entomology. It deals with the basic or academic aspects of the science of entomology. It includes morphology, anatomy, physiology and taxonomy of the insects. In this case we study the subject for gaining knowledge on entomology irrespective of whether it is useful or harmful. Applied entomology or economic entomology. It deals with the usefulness of the science of entomology for the benefit of mankind. Applied entomology covers the study of insects which are either beneficial or harmful to human beings. It deals with the ways in which beneficial insects like predators, parasitoids, pollinators or productive insects like honeybees, silkworm and lac insect can be best exploited for our welfare. Applied entomology also studies the methods in which harmful insects or pests can be managed without causing significant damage or loss to us. Economic classification of insects. Insects can be classified as follows based on their economic importance. 1. Insects of no economic importance. There are many insects found in forests and agricultural lands which neither cause harm nor benefit us. 2. Insects of economic importance. A. Injurious insects. A. Pests of cultivated plants, crop pests. Each cultivated plant damaged by many insect pests which feed on them reduces the yield of the crop. E.g. cotton bollworm, rice stem borers. B. Storage pests. Insects feed on stored products and cause economic loss. E.g. rice weevil, pulse beetle. C. Pest attacking cattle and domestic animals. Cattle are affected by pests like horsefly, flesh fly, fleas and lice. They suck blood and sometimes eat the flesh. D. Household and disease-carrying insects. Household pests include cockroach, ants, etc. Disease-carrying insects are like mosquitoes, houseflies, bed bugs, fleas, etc. 6. B. Beneficial insects. I. Productive insects. I. Silkworm. The silkworm filament secreted from the salivary gland of the larva helps us in producing silk. E. Honeybee. Provides us with honey and many other byproducts like beeswax and royal jelly. E. Lac insects. The secretion from the body of these scale insects is called lac. Useful in making vanishes and polishes. IV. Insects useful as drugs, food, ornaments, etc. As medicine, e.g. sting of honey bees remedy for rheumatism and arthritis. Enthoradin. Extracted from blister beetle useful as hair tonic. B. As food for animals and human being. For animals, aquatic insects used as fish food. Grasshoppers, termites, pupae of moths. They have been used as food by human beings in different parts of the world. C. Ornaments, entertainers. Artists and designers copy color of butterflies. Beetles worm as necklace. Insect collection is a hobby. D. Scientific research. Drosophila and mosquitoes are useful in genetic and toxicological studies respectively. 2. Helpful insects. I. Parasites. These are small insects which feed and live on harmful insects by completing their life cycle in a host and kill the host insect. E.g. egg, larval and pupal parasitoids. E. Predators. These are large insects which capture and devour harmful insects. E.g. coccinellids and praying mantid. E. Pollinators. Many cross-pollinated plants depend on insects for pollination and fruit set. E.g. honey bees, aid in pollination of sunflower crop. IV. Weed killers. Insects which feed on weeds kill them, thereby killers. E.g. Mexican beetle. Eats on Parthenium, Gajar gavet. Cochineal insect feeds in Apuntia dilloni. V. Soil builders. Soil insects such as ants, beetles, larvae of cutworms, crickets, columbola, make tunnels in soil and facilitate aeration in soil. They become good manure after death and enrich soil. V. Scavengers. Insects which feed on dead and decaying matter are called scavengers. They important for maintaining hygiene in the surroundings, e.g. carrion beetles. Rove beetles feed on dead animals and plants. D. Household and disease-carrying insects. I. Pests which cause damage to belongings of human being like furniture, wool, paper, etc. E.g. cockroaches, furniture beetle, sliverfish, etc. E. Pests which cause painful bite, inject venoms. E.g. wasps, bees sting us. Hairy caterpillar. Nettling hairs are poisonous. Mosquitoes, bugs bite, piece and suck blood from us. E. Disease-causing mosquito malaria, filariasis, dengue fever, housefly typhoid, cholera, leprosy, anthrax. 7. 8. Insect dominance. Measures of dominance. 1. More number of species. 2. Large number of individuals in a single species, e.g. locust swarm comprising of 10. 9. Numbers of individuals, occupying large area. 3. Great variety of habitats. 4. Long geological history. Reasons for dominance. There are several structural, morphological and physiological factors responsible for insect dominance. They are 1. Capacity for flight. 2. More adaptability or universality. Smaller size. Majority of insects are small in their size, conferring the following physiological and ecological advantages. 3. Presence of exoskeleton. Insect body is covered with an outer cuticle called exoskeleton, which is made up of a cuticular protein called chitin. This is light in weight and gives strength, rigidity and flexibility to the insect body. 4. Resistance to desiccation. Insects minimize the water loss from their body surface through prevention of water loss. Wax layer of epicuticle, closable spiracles, eggshell. Conservation of water, capable of utilizing metabolic water, reabsorption of water from fecal matter, use less quantity of water to remove the nitrogenous waste. 5. Tracheal system of respiration. This ensures direct transfer of adequate oxygen to actively breathing tissues. Spiracles through their closing mechanism admit air and restrict water loss. 6. Higher reproductive potential. Reproductive potential of insect is high, e.g. egg-laying capacity, fecundity, of queen termite is 6,000-7,000 eggs per day for 15 long years. Short development period, e.g. corn aphid produces 16 nymphs per female, which reaches the adulthood within 16 days. Presence of special types of reproduction other than oviparity and viviparity like polyembryony, parthenogenesis and poidogenesis. 7. Presence of complete metamorphosis. More than 82% of insects undergo complete metamorphosis, holometabolous insects, with 4 stages. As the larval and adult food sources are different, competition for food is less. 8. Presence of defense mechanisms. By different defense mechanisms, insects escape from the enemies to increase their survival rate. 9. Hexapod locomotion. Insects uses 3 legs at a time during locomotion, while the remaining 3 legs are static, which gives greater stability. Insect integument. The vertebrates have internal skeleton known as endoskeleton, while in insects it is located outside the body, forming exoskeleton. Insect cuticle provides space for attachment of muscles of antenna and mouthparts, called as tentorium. Insect body wall is called as integument or exoskeleton. It is the external covering of the body which is ectodermal in origin. It is rigid, flexible, lighter, stronger and variously modified in different body parts to suit different modes of life. Integument consists of 3 layers. 1. Cuticle, upper. 2. Epidermis or hypodermis, middle. 3. Basement membrane, inner. 1. Cuticle. It is outermost thick layer of integument secreted by epidermis. It is non-cellular. It is divided into 2 regions, epicuticle upper and procuticle inner, a epicuticle. It is a thin outermost layer varying in thickness from 1 to 4 mu. Chitin is absent in epicuticle. It consists of the following 4 layers. 9. Cement layer. It is secreted by dermal glands and is composed of lipoprotein. It protects the body from external damage. Wax layer. It consisting of long-chain hydrocarbons, esters of fatty acids and alcohols. It serves as waterproof layer, preventing water loss from the body. Polyphenol layer. It is a non-static layer containing various types of phenols which are mainly used in the formation of the proteins. It is resistant to acids and organic solvents. Cuticulin layer. It is an amber-colored thin layer over the surface of the epidermis which is strengthened by outer polyphenol layer. It serves the purpose of permeability and also acts as growth barrier. B. Procuticle. It is differentiated into exo and endocuticle. Exocuticle. It is darkly pigmented, hard and sclerotized. This layer is made up of chitin and sclerotin. Endocuticle. It is soft, light-colored and unsclerotized. This layer is made up of chitin and arthropodin. Pore canals. These are numerous fine vertical channels traversing both exo and endocuticle. Pore canals present in the exocuticle helps in the deposition of epicuticle. They are useful in transportation of cuticular material and enzymes to the outer pro and epicuticle parts. Composition of cuticle. Club suit. 2 major components of insect cuticle are I. Chitin, E. Proteins. I. Chitin. It is a nitrogenous polysaccharide. Composition of cuticle. Club suit. Insect cement layer. It is secreted by dermal glands and is composed of lipoprotein. It protects the body from external damage. Wax layer. It consisting of long-chain hydrocarbons, esters of fatty acids and alcohols. It serves as waterproof layer, preventing water loss from the body. Polyphenol layer. It is a non-static layer containing various types of phenols which are mainly used in the formation of the proteins. It is resistant to acids and organic solvents. Cuticulin layer. It is an amber-colored thin layer over the surface of the epidermis which is strengthened by outer polyphenol layer. It serves the purpose of permeability and also acts as growth barrier. B. Procuticle. It is differentiated into exo and endocuticle. Exocuticle. It is darkly pigmented, hard and sclerotized. This layer is made up of chitin and sclerotin. Endocuticle. It is soft, light-colored and unsclerotized. This layer is made up of chitin and arthropodin. Pore canals. These are numerous fine vertical channels traversing both exo and endocuticle. Pore canals present in the exocuticle helps in the deposition of epicuticle. They are useful in transportation of cuticular material and enzymes to the outer pro and epicuticle parts. Composition of cuticle. Club Soot's two major components of insect cuticle are I. chitin, E. proteins. I. chitin. It is a nitrogenous polysaccharide. It consists of high molecular weight polymer of anhydro-N-acetyl-glucosamine residues joined by beta-glycosidic linkages. It is water-insoluble but soluble in dilute acids, alkalis and organic solvents. E. proteins. Cuticle has three types of proteins. Arthropodin. An untanned protein means it is soft water-soluble protein present in endocuticle. The conversion of arthropodin into sclerotin is known as sclerotization or tanning. Sclerotin. It is also called tan protein, which is amber-colored and present only in exocuticle. This is water-insoluble. Resilin. It is a rubber-like elastic protein which is colorless and present in joints such as wing hinge ligaments, leg joints, clipolabral joints or suture and tergosternal joints. 10. 2. Epidermis or hypodermis. It is an inner unicellular layer resting on basement membrane with the following function. I. Cuticle secretion. E. Digestion and absorption of old cuticle. E. Wound repairing. IV. Gives surface look. Adjacent epidermal cells are held together by means of certain cytoplasmic processes which are known as Adjacent epidermal cells are held together by means of certain cytoplasmic processes which are known as desmosomes. All the epidermal cells are glandular and secrete cuticle and the enzymes involved in production and digestion of old cuticle during molting. The epidermal cells get differentiated into following types based on the function they perform and may modify into A. Dermal glands producing cement layer. B. Trichogen cell producing hair-like seta or trichome. C. Molting glands secreting molting fluid which digests the old cuticle. D. Peristigmatic glands around the spiracles in case of dipteran larva. 3. Basement membrane. It is the basal part of the body wall formed from degenerated epidermal cells and appears as non-living amorphous, shapeless, granular layer of integument. It is about 0.5 micro in thickness and consists of fibrous protein, glycosaminoglycans which are polymers of disaccharides. The basement membrane forms a continuous sheet beneath the epidermis, where muscles are attached and become continuous with sarcolemma of the muscles. Functions of insect integument. O. It provides protection to the internal organs enclosed by it. O. It gives shape and size to the insects. O. It provides surface for the attachment of muscles. O. It conserves moisture and prevents desiccation. O. It prevents entry of pathogens and insecticides. O. It forms sense organ. O. It contains pigments to make insects attractive. 11. 12. Cuticular modifications. Cuticular modification. A. Cuticular outgrowths. They are divided into cuticular appendages and cuticular processes depending on the presence or absence of membranous articulations. Cuticular appendages. These are the outgrowths of the cuticle, integument connected with it by means of a membranous joint. They arise from modified epidermal cells. These are classified into setae and spurs. 1. Seta, macrotrichia. Club Soot commonly known as hairs and arise from a cup-like alveolus or pit. Setae are hollow structures developed as extension of exocuticle and are produced by a single enlarged hypodermal cell called trichogen cell. Articular membrane is usually produced by a second hypodermal cell called tormogen cell. Club Soot setae have role of taxonomic importance and vary with species to species. Study of arrangement of setae is known as chaetotaxy. 2. Spurs. Club Soot occur on the legs of many insects and differ from setae in being multicellular in origin. 2. Cuticular processes. They have no membranous articulation. They are of two types. 1. Microtrichia, fixed hairs, aculae. Club Soot. These are minute hair-like structures found on wings of Mecoptera and certain Diptera. 2. Spines. Club Soot outgrowths of the cuticle which are more or less thorn-like in form. Cuticular outgrowths. Cuticular invagination. Apodems. Apophyses. Cuticular appendages. Cuticular processes. Setae spurs. Aculae spines. 13. Senior. No. Spurs spines. 1. Cuticular appendages. Cuticular processes. 2. Movable, multicellular structures and thick. Walled. These are immovable outgrowths of cuticle. 3. E.g. present on tibia of plant hoppers and honeybees. E.g. hind tibia of grasshopper and leaf hoppers. B. Cuticular invagination. The body wall or cuticle of the body wall invaginate internally and grow into definite structures which are of two types. Spade Soot apodem. Shallow cuticular invaginations which provide area for muscle attachment. Spade suit apophysesolid invaginations of the cuticle which gives mechanical support to various organs by forming distinct Spade suit apophysesolid invaginations of the cuticle which gives mechanical support to various organs by forming distinct skeletal structures. Molting process. Process of periodical shedding of old cuticle and formation of new cuticle. This process is known as molting. Itis, a complex process which involves apolysis, ecdysis and sclerotization. Apolysis. Apolysis. Apo equals formation, lysis equals dissolution. The dissolution of old cuticle and formation of new one is known as apolysis. It starts with repeated meiotic division of epidermal cells resulting in increase in number and size of epidermis. Because of this change, the epidermal cells exert tension on cuticular surface and as a result get separated them from the cuticle. Due to separation of epidermis from the cuticle, a subcuticular space is created and the epidermal cells starts producing their secretion, i.e. molting fluid and cuticular material into this space. Ecdysis. The stage. Itis, a complex process of periodical shedding of old cuticle and formation of new cuticle. This process is known as molting. Itis, a complex process which involves apolysis, ecdysis and sclerotization. Apolysis. Apo equals formation, lysis equals dissolution. The dissolution of old cuticle and formation of new one is known as apolysis. It starts with repeated meiotic division of epidermal cells resulting in increase in number and size of epidermis. Because of this change, the epidermal cells exert tension on cuticular surface and as a result get separated them from the cuticle. Due to separation of epidermis from the cuticle, a subcuticular space is created and the epidermal cells starts producing their secretion, i.e. molting fluid and cuticular material into this space. Ecdysis. The stage where the insect has both newly formed EPI and procuticle. The ecdysial membrane starts splitting along the line of weakness due to muscular activity of the inner developing insect and also because of swallowing of air and water resulting in the distension of the gut and also due to the pumping of blood from abdomen to thorax through muscular activity. After the breakage of old cuticles, the new instar comes out bringing its head followed by thorax, abdomen and appendages. Sclerotization. After shedding of old cuticle, the new cuticle which is soft, milky white colored becomes dark and hard through the process known as tanning or sclerotization. 3 types of hormones involved in the process of molting which are as follows. JH, juvenile hormone, produced from corpora allata of brain that helps the insects to be in immature stage. MH, molting hormone, produced from prothoracic glands of brain that induces the process of molting. Eclosion hormone, released from neurosecretory cells in the brain that help in the process of ecdysis or eclosion. Body segmentation of insect. Cockroach, Paraplanata americana is a typical insect as it possesses all important characters of class insect. In general, insect body is divided into a series of rings or segments are known as somites or metameres. During the process of evolution, these somites get fused with each other in different ways forming the body parts of the existing arthropods. The type of arrangement of these body segments in embryonic stage is known as primary segmentation while in adult insects is known as the secondary segmentation. Insect body is divided into 3 regions or tagmata, namely head, thorax and abdomen. This grouping of body segments into regions is known as tagmosis. Head consists of 6 segments, possesses mouthparts, compound eyes, simple eyes, ocelli, and a pair of antennae. Thorax consists of 3 segments, i.e. prothorax, mesothorax and metathorax. Meso and metathorax are together known as pterothorax. All the 3 thoracic segments possess a pair of legs and meso and metathorax possess 1 pair of wings. Abdomen has 11 segments with genital appendages on 8th and 9th segments. The insect body generally consists of 20 segments. Figure general organization of insect body. 14. 15. Insect head. It is the foremost part in insect body consisting of 6 segments that are fused to form a head capsule. The head segments can be divided into 2 regions, i.e. procephalon and gnathocephalon, mouth. Head is attached or articulated to the thorax through neck or cervix. Head capsule is sclerotized and the head capsule excluding appendages formed by the fusion of several sclerites is known as cranium. Inside the head, an endoskeletal structure called the tentorium which gives supports to the brain, and provides a rigid origin for muscles of the mandibles and other mouthparts. Head is concerned with feeding and sensory perception. Segment appendages. I pre-antennary segment procephalon, pair of compound eyes and 3 ocelli, simple eyes, 2 antennary segment, pair of antennae, 3 intercalary segment, single labrum, IV mandibular segment, gnathocephalon, pair of mandibles, V maxillary segment, pair of maxillae, V labial segment, single labium. Types of head position. The orientation of head with respect to the rest of the body varies. According to the position or projection of mouth parts, the head of the insect can be classified as Up hypognathus, hypo, below, gnathus, jaw. The head remain vertical and is at right angle to the long axis of the body and mouth parts are ventrally placed and projected downwards. This is also known as orthopteroid type, e.g., grasshopper, cockroach. Cockroach grasshopper. B. Prognathus, pro, in front, gnathus, jaw. The head remains in the same axis to body and mouth parts are projected forward. This is also known as coleopteroid type, e.g., beetles. Beetles. C. Opistognathus, opisto, behind, gnathus, jaw. It is same as prognathus but mouth parts are directed backward and held in between the forelegs. This is also known as hemipteroid or opisthorinchus type, e.g., bugs. 16. Mosquito red cotton bug. Sclerites and sutures of head. The head capsule is formed by the union of number of sclerites or cuticular plates or areas. The head capsule is formed by the union of number of sclerites or cuticular plates or areas which are joined together by means of cuticular lines or ridges known as sutures or any of the large or small sclerotized, hardened areas of the body wall. These sutures provide mechanical support to the cranial wall. Suture. The sclerites separated from each other by means of thin impressed line called suture. Sometimes referred as a sulcus. General insect possess the following sclerites. 1. Labrum. It is small sclerite that forms the upper lip of the mouth cavity. It is freely attached clypeus by means of clipolabral suture. 2. Clypeus. It is situated above the labrum, separated by frontoclypeal suture and also separated from genna by clipogenal suture. 3. Frons. It is unpaired, facial part of the head capsule lying between the arms of epicranial suture. 4. Genna. It is the area extending from below the compound eyes to just above the mandibles. It is separated from frons. It is freely attached clypeus by means of clipolabral suture. 2. Clypeus. It is situated above the labrum, separated by frontoclypeal suture and also separated from genna by clipogenal suture. 3. Frons. It is unpaired, facial part of the head capsule lying between the arms of epicranial suture. 4. Genna. It is the area extending from below the compound eyes to just above the mandibles. It is separated from frons by frontogonal suture and from clypeus clipogenal suture. 5. Epicranion. It forms the upper part of the head extending from frons to the neck. 17. 6. Vertex. It is the top portion of epicranium which lies behind the frons or the area between the two compound eyes. 7. Epicranial suture. Starting from the dorsal portion of the epicranium is an inverted Y-shaped suture known as epicranial suture or ecdysial line. The head capsule breaks open along this line at the time of molting. 8. Occiput. It is an inverted, U-shaped structure representing the area between the epicranium and post-occiput. 9. Post-occiput. It is the extreme posterior part of the insect head that remains before the neck region. 10. Ocular sclerites. These are cuticular ring-like structures present around each compound eye. 11. Antennal sclerites. These form the basis for the antennae and present around the scape. Anterior view or face view. The common sutures present in head are 1. Clipolabral suture. It is the suture present between clypeus and labrum. 2. Clipofrontal suture or epistomal suture. The suture present between clypeus and frons. 3. Epicranial suture. It is an inverted, Y-shaped suture distributed above the facial region extending up to the epicranial part of the head. It consists of two arms called frontal suture occupying the frons and stem called as coronal suture. This epicranial suture is also known as line of weakness or ecdysial suture because the exuvial membrane splits along this suture during the process of ecdysis. 4. Occipital suture. It is U-shaped or horseshoe-shaped suture between epicranium and occiput. 18. 5. Post-occipital suture. It is the only real suture in insect head. Posterior end of the head is marked by the post-occipital suture to which the sclerites are attached. As this suture separates the head from the neck, hence named as real suture. 6. Genal suture. It is the sutures present on the lateral side of the head, i.e. genna. 7. Ocular suture. It is circular suture present around each compound eye. 8. Antennal suture. It is a marginal depressed ring around the antennal socket. Eight. Antennal suture. It is a marginal depressed ring around the antennal socket. Insect thorax. It is the middle part of the body consisting of three segments: is prothorax, mesothorax and metathorax, each possessing a pair of legs and a pair of wings on meso and metathoracic segment. Meso and metathoracic segments bear a pair of wings each together known as pterothorax. Ptera equals wings. Thorax generally concerned with locomotion. The body wall of a typical insect is divided into four regions. The dorsal upper region is called dorsum or tergum notum. The ventral lower region is called as venter or sternum. The two lateral regions are known as pleury pleuron. Sclerites. The cuticle hardens at localized areas form sclerites. 19. 20. Sutures. The sclerites are separated from each other by means of thin impresses lines called as sutures. Sclerites forming these regions are called as tergites, sternites and pleurites, respectively. Sclerites of thoracic segments. 1. Sclerites of tergum. Tergites. The dorsal sclerites consists of three segmental plates, nota called pronotum, mesonotum and metanotum. Each notum is again divided into three parts, i.e. pre-scutum, scutum and scutellum. 2. Sclerites of pleuron. Pleurites. It is fully developed in winged insects. It is divided into two parts, anterior episternum and posterior epimeron. It is absent in prothorax. 3. Sclerites of sternum. Sternites. It is divided into eusternum and spinasternum. Insect abdomen. The abdomen in the embryo usually consists 11 segments. The abdominal segments are sometimes designated as uromers. The terminal region of abdomen is called telson, which bears anus. The first abdominal segment gets fused to metathorax, forming propydium. In ants, bees and wasps. The first 8 abdominal segments carry a pair of spiracles each. Thorax generally concerned with reproduction and metabolic activity. Appendages of abdomen. 1. Non-reproductive appendages. Of cerci, they are present on 11th segment in most of the insects. It is present in male cockroach, silverfish, grasshopper. Cerci usually act as tactile organ or sound receptors in grasshopper. They become a part of male genitalia in caddisfly. In earwigs, cerci are modified into defensive organ. B. Prolegs. In insect larvae. Larvae of Lepidoptera bear 5 pairs of abdominal legs called prolegs on 3rd, 4th, 5th, 6th and 10th segments. These prolegs bear spines-like structures called crochets, on terminal ends to grip the plant surfaces. In case of larvae of sophli, there are 8 pairs of prolegs but are without crochets. C. Abdominal gills. It is present in aquatic insects for respiration, e.g. nymph of Odonata. D. Cornicles. Aphids have a pair of short tubes known as cornicles or siphonculae projecting from dorsum of 5th or 6th abdominal segment. They permit the escape of waxy fluid, which perhaps serves for protection against predators. 2. Reproductive appendages. It includes abdominal segments from 1 to 7 or progenital segments. 8th and 9th are known as genital segments as they form genital appendages, i.e. ovipositor in females and edagus or penis in males. 10th and 11th segments are known as postgenital segments. These organs are specially concerned with mating in male and deposition of eggs in females. They are collectively called as external genitalia or gonapophysis. Male external genitalia. The 9th sternum bears 2 styli and pair of claspers which help to hold female during copulation. The edagus lies between claspers. Female external genitalia. It has a special egg-laying organs called ovipositor for egg lying on 8th and 9th segments. The ovipositor of housefly and fruit fly is called pseudovipositor.